Okay, in this video, we're going to talk about digestion and absorption. So, digestion is a process whereby we break down the food you eat into from large chunks into smaller pieces by mechanical digestion, and then further break it down by chemical digestion into absorbable nutrients. Later on, we'll talk about the regulation of the digestive process, but in this video, we're going to specifically talk about absorption of digested nutrients. So, absorption of nutrients is actually not highly regulated, meaning that you will absorb everything you eat as long as you have a mechanism to absorb those nutrients. So we'll talk about the three main classes of macromolecules and their digestion and absorption. So carbohydrates, lipids, and proteins, and of course water. And absorption of water is very important. So let's start out by just sort of orienting ourselves to what's on this drawing here. And you have a copy of this notes template, so you should follow along with this and draw along with me. So here we've got some blood. And these cells that are right up here are intestinal cells, and in fact, they're small intestine cells, so the duodenum. So I'll just make a note that these are duodenal cells. And on this side of the duodenum cells is what we call the lumen. So this is where the digested material would be entering that small intestine or duodenum. So what we're going to do is list the three major classes of carbohydrates, what digests them, and what type of enzymes, and the monomers that they're digested into, and the absorption mechanism of those. So let's start out with carbohydrates. And again, we're just going to hit the highlights of this. We're not going to include an exhaustive list of possible enzymes and monomers. So starch is, or the main carbohydrate that you eat likely is starch, the storage form of carbohydrate in plants. And carbohydrates are digested into mono and diglycerides typically. So single and double sugars. And a good example of a monoglyceride is glucose. I'm going to show the enzymes in green. The enzyme that typically breaks down starch is amylase, which you probably already knew that. And amylase is present in both your saliva and in your pancreas. So you actually start breaking down starch in your mouth. And to test this, you can put a cracker in your mouth, and if you let it sit there with a bunch of saliva, it'll start getting sweeter over time. Kind of a gross experiment, but it gets the point across. So let's do one other class of carbohydrates. Disaccharides have to be further broken down. So to give you an example of how you can sometimes figure out enzyme names, disaccharides are broken down into monosaccharides, and the enzyme that breaks down disaccharides is called a disaccharidase. So usually you can figure out what an enzyme does because it has the name of what it breaks down with ASE on the end of it. So disaccaridases break down disaccharides. Okay, so how do we absorb? Let's talk about glucose to start with. So how do we absorb glucose? So let me circle glucose here. I'm going to do my transport mechanisms in blue. So you guys already know this. It is exactly the same process that occurs in the as what occurs in the kidney. And so I'm just making note that we have interstitial fluid here and intracellular fluid here, just so we're clear about that. Okay, so the first thing for absorption of glucose that we have to have is the sodium potassium ATPase, always working on what we call the basal side of the membrane here. And you guys probably remember that sodium is pumped out in exchange for potassium. And of course, lots of ATP is used to do this. So that keeps sodium levels really, really low in there. And I'm going to try to draw an itty bitty sodium, and there's lots more sodium out here. And so that sodium gradient then allows for the secondary active transport of glucose. So we'll actually move glucose against its concentration gradient by sodium following its concentration gradient. The energy in that gradient allows glucose to be moved in against its gradient. So I'm going to draw a little bit of glucose here and more glucose here. So now we've got high levels of glucose inside the cell. We've moved it in by secondary active transport, so that's against its concentration gradient. Well, then we can move it out by facilitated diffusion. And remember, we talked about the fact that it's diffusion because it's following its concentration gradient, and it's facilitated diffusion because it needs a protein transporter in the membrane because it's not soluble. And then, of course, that stays low in this interstitial fluid here because it's picked up right away by the blood. Okay, so nothing new there. You guys already knew how glucose was absorbed. So let's talk about proteins. So proteins, we'll put them down here. They are broken down into, we'll say, peptides, which are pieces of proteins, and amino acids. And we're going to be using the abbreviation for an amino acid. So let me write amino acid one time, and then forevermore we will abbreviate it AA. So lots of different enzymes can break proteins down into peptides and amino acids. So let's just put a few of them down here. We have proteases and peptidases. So again, you would imagine a protease would break apart proteins. And a peptidase, you might imagine, would make peptides smaller. And it does, breaks down peptides into amino acids. And most of these are from the small intestine or from the pancreas. So small intestine or pancreas. So those are from the exocrine cells of the pancreas. However, there's one enzyme that comes from the stomach, so that the breakdown of protein starts before it even gets into the stomach, and that's pepsin. So put pepsin from the stomach. And you may recall that pepsinogen is the precursor form of pepsin. It's converted to pepsin when it hits the acid of the stomach. So it's pepsinogen inside the cells that make it, and then when it gets released into the stomach, the lumen of the stomach, the low pH of like about 2 converts it into pepsin. The general name for a precursor enzyme is a zymogen. That's the name of an enzyme that's not active yet. And in fact, all of the enzymes, until they're released, are referred to as zymogens. They become active once they're released into the space where they're going to be used. Otherwise, they would digest the cells that made them, which wouldn't be very good. Okay, how do we absorb amino acids? So let's look at this cell here. Again, we have to have the sodium potassium ATPase making a gradient for us, a sodium gradient. So this is the same one we had over in our glucose cell. So the sodium potassium ATPase sets up that gradient, which allows for, just like with glucose, the secondary active transport of amino acids. So we've got amino acids, which I'll abbreviate as AA, and they're often and they're co-transported to get in with sodium. So sodium's following its concentration gradient. So remember, there's lots more out here. Well, then amino acids actually can leave the cell in different ways. One way is co-transport with hydrogen ions. So exchange. And there's a different secondary active transporter for most of the different amino acids. So it's a very similar type of absorption as glucose. But the point is, it's a specific transporter requiring energy in the form of ATP that is hydrolyzed in this process of active transport here, allowing for secondary active transport of these molecules up here. There is another way that peptides can be absorbed, and that's through the process of transcytosis, which is why I left this cell the way it is. And that is, we can have endocytosis of pieces of proteins. So I'm going to draw some, these are peptides right here. We would never absorb a whole protein because by the time they get into the small intestine, they're broken apart anyway. And then we have them as a vesicle in here, and then they will leave by exocytosis. So here we've got that vesicle moving across the cell, and then these peptides are released by exocytosis. So this whole process is called transcytosis, where we have endocytosis of these peptides. They move across the cell, so it's a directional movement, and then we have exocytosis of these on the other side. This doesn't really happen too much in adult digestive tracts, a little bit more so in newborns, which is why you have to be careful about what you feed babies, especially in the form of proteins when they're really young, because it's believed that absorbing these partial proteins can maybe lead to problems with allergies later on. Okay, so, but again, not too much of this happening in the adult digestive tract. There's too much infolding, and the structure of it's not, doesn't allow too much of that to happen. So the last class of macromolecules I wanted to talk about was fats, or lipids. And fats are taken in as large blobs of fat. Very technical term here, but it's kind of right. And those large blobs are emulsified into small blobs, which more technically are referred to as micelles. And what causes this emulsification is bile salts, and they kind of work a little bit like soap does. So if you have greasy dishes in soapy dishwater, the large areas of fat are spread out, and they become soluble, not really soluble, but emulsified in the water, so that the amphipathic soap molecules, which are polar on one side and nonpolar on the other, allow those fats to be spread out in the dishwater and removed from the plates. And this increases the surface area of the fats so that they can be digested by lipases into fatty acids and monoglycerides. And that was lipases that digest that. So the emulsification is necessary. I'm going to write emulsify here. Is necessary to increase the surface area of the fats so that they are available to the lipases to be efficiently digested. So how do fatty acids get into cells? Well, remember they're fat soluble, so they are able to just move in by simple diffusion. And you know that this is simple diffusion because there's no carrier protein in the membrane here. Cholesterol is, despite the fact that it's a lipid, enters the cell by some type of carrier-mediated transport. So cholesterol. And within the cell, the cholesterol, the fatty acids, which I need to label those on there. So fatty acids, we're going to abbreviate as FA. So FA for fatty acids. And so these are fatty acids diffusing in here. So the fatty acids and the cholesterol combine with protein to form a structure inside the cell called a chylomicron. So we have fatty acids, cholesterol, I'm going to abbreviate cholesterol there, and protein. I'm going to abbreviate protein. So fatty acids, cholesterols, and proteins form this structure called a chylomicron, which is too big to cross the membrane. So it actually leaves the cell by exocytosis. So that's our chylomicron right there. So I'm going to put the stripes on this one so we know it's the same thing. So this chylomicron leaves the cell by exocytosis, and it can't get into the blood vessels. Again, it's a little bit too big. And so it enters lacteals, which are part of the lymphatic system. And this is how fats travel. They enter the lymphatic system and then they get dumped back into the blood at the vena cava, just like when we talked about lymphatic fluid forming. But remember, fats are not soluble in water, so they have to be carried around with proteins and cholesterol as well, and this is what we call lipoproteins. And these lipoproteins, that are these mixtures of fatty acids and cholesterol and protein, are the low density lipoproteins that end up traveling to your liver. Okay, we left out one important thing, which was water. So how is water absorbed across your digestive tract? Well, water has to follow solutes. So water in the lumen here will move in either between cells or across water channels or aquaporins following the absorption of these solutes. So it moves across and then into the blood. So water follows solutes. And remember when we talked about the sugar-free gummy bears, that if you have unabsorbable solutes in this lumen space like glycosan or olestra or other solutes you just don't have an absorption mechanism for, water will be held back because the concentration gradient between the lumen and the inside of these cells is lower, so you don't absorb as much water if you have unabsorbable solutes. So that's why the sugar-free gummy bears gave you diarrhea. Okay, so make sure that you know these transmembrane trafficking mechanisms. You know secondary active transport, you know primary active transport, facilitated diffusion, and simple diffusion, and know how these monomers of these molecules are able to be absorbed in this way. And again, if you don't have a specific absorption mechanism or protein in the membrane for a molecule, then you cannot absorb it.